First of all, we observe the confluence of our fibroblasts using an inverted phase contrast microscope. To start, we wash the cell mat, we remove all the medium present in the T25 and then rinse the mat with 10 ml of PBS, which is then discarded. Then the cell mat is detached by adding 1 ml of trypsin, incubated at 37 degrees for 1 to 4 minutes, depending on the confluence of the cell mat. The cells are gently tapped to loosen and suspend. We observed our cells under the IPCM. To inactivate trypsin, we add 5 ml of medium. The suspension is homogenized and returned to a falcon tube, breaking up the cell clusters. The cells are then counted in a cover slide to determine the number of cells in the suspension. We observed at 100 times magnification. We take the volume corresponding to 2 times 10 to the 5th power cells per milliliter, according to our count. Then we centrifuge at 1500 G. The supernatant is removed and the cell pellet is recovered in 2000 microliter of pure SVF. We place 1600 microliter of type 1 collagen in a falcon tube. We add then 17 microliter of NaOH and 2000 microliter of HBSS and mix well. Then we add 2000 microliter of fibroblast solution and mix well. Then we place an insert in 2 wells on a 12 plate at 750 microliter to each insert and incubate the plate for 1 hour at 37 degrees to allow the collagen to polymerize. During this time, we prepare a control solution of 1 ml of medium with 1.5 microliter of cholera toxin, with 1 microliter of hydrocortisone, with 1 microliter of ascorbic acid. We prepare an identical test solution by not adding what you wish to test to the tube. Hydrocortisone is not deposited here. Then we remove the plate and place the solution prepared above on the polymerized collagen layer in the control well and in the test solution in the other insert. Then we incubate again this plate. First, we observe the confluence of our keratinocytes at IPCM, then we wash the cell layer, all the medium present in the T25 is removed. Then we rinse the layer with 10 ml of PBS, which is discarded afterwards. The cell layer is detached by adding 1 ml of trypsin and incubated at 37 degrees for 1 to 4 minutes, depending on the confluence of the cell layer. We gently tap to detach the cell to put them into suspension. To inactivate the trypsin, we add 5 ml of medium. The suspension is homogenized and transferred to a falcon tube, ensuring that the cell clumps are properly broken up. A cell count is performed on the cover slide to determine the number of cells in the suspension. We observe this at 100 times magnification under an optic microscope. We take the volume corresponding to 5 times 10 to the 5th power cells per milliliter according to our count and prepare 2 falcon tubes, the test tubes and the control tubes. Then we centrifuge the tubes. During this time we prepare the control tube with 1 ml of medium is added with 1.5 microliter of cholera toxin. with 1 microliter of hydrocortisone and 1 microliter of ascorbic acid. For the test tube, a test solution identical to the control is prepared, except that the substance we wish to test, in this case hydrocortisone, is not added. After the centrifugation, the supernatant is discarded and the cell pellet is taken up with the control solution and the test solution. The plates are taken, the medium present on the collagen lath is removed, and 500 microliter of cell suspension taken from the control and test solutions is put on the collagen lath. Then we incubated the plate at 37 degrees during 24 hours. First, we take the plate from the incubator, we take the insert and we carefully cut the membrane of the insert and place the sample in a clean cassette. The cassette is closed and immersed in 4% formaldehyde. We take the cassette and place it in a beaker. We wash 2 times of 5 minutes in PBS under agitation. The beaker is emptied into a recovery container. We perform a 70% ethanol bath during 30 minutes. Then we perform a 95% ethanol bath for 13 minutes. Then we perform 3 times an absolute ethanol bath for 30 minutes. For the last one, we incubate 1 hour. We perform a new xylem bath for 30 minutes and another one with fresh xylem for 1 hour. Next, a paraffin bath at 58 degrees is performed for 30 minutes. Melted paraffin is poured at the bottom of the mold. The sample is placed in the correct orientation. Covered with paraffin, the cassette is replaced and the mold is filled with paraffin. The paraffin solidified in 30 minutes. The paraffin block containing the sample and then unmolded. Then the cassette is placed on the microtome, the blade is advanced and the paraffin embedded sample is cut. The sections are collected with tweezers and placed in a warm water bath. The samples are then collected on slides. The slides are put in different baths, a xylem bath for 10 minutes and repeat with a fresh bath for 13 minutes, an absolute ethanol bath for 3 minutes 2 times, a 95% ethanol bath for 1 minute, a 80% ethanol bath for 1 minute, a distilled water bath for 3 minutes, a hematoxylin bath for 30 seconds, a tap water bath for 3 minutes, an aqueous eosin Y bath for 30 seconds, and then a distilled water bath for 30 seconds. Then the slides are dried and they are observed under an optic microscope.